Okay, so those are the three modes of specification. Again, conditional. If you move cells from one spot to another, and they will respond differently to a different area based upon the signals, the morphogen gradients, and the cell-to-cell signaling that goes on in that particular area. Now, there comes a point where the cells become determined. Remember, when they're determined, when you move them to another place, they no longer respond to various signals that are already established in their fate. So in that regard, conditional specification creates these patterns within the embryo, but only to a certain point, and then the cells are going to, for the most part, become specified autonomously because they're already specified in their fate, or determined, I should say. All right, so let's talk about induction and competence, because this is an important part of conditional specification. Okay, so mostly what we're going to talk about right now is conditional specification and some of the factors behind it, since most of what we study is for the benefit of humankind, and therefore we want to look at the method of our development so we can understand stem cell therapy and other things a little bit better for us. So induction and competence. Let's define these words first and then give you some examples of how this works. When something is induced, it ultimately produces a signal, or the induction process is kind of the morphogen gradients. Ultimately, these cells produce a signal, a protein, that can change the behavior of adjacent cells. So the inducer are the cells that are producing the protein, the morphogen. So induction is just the process of one cell telling another cell what to do based upon its secretion of its proteins. Or, as we'll show, sometimes just it doesn't actually secrete the proteins. It just kind of has some membrane proteins that will touch the other cell and still tell it what to do. Now, just because one set of cells is sending out a signal does not mean that the other cells have the capacity to respond to that. That capacity is what we call competence. So competence is the ability for the cells to be able to respond to the inducting signal. Okay? Early on, most cells are competent. But as the cells become determined, as they become determined in what fate they're going to become, their competency goes to almost zero, meaning they don't respond to any of the adjacent cells. They're already determined in what fate they're going to become. So really, induction and competence are in the earliest stages of specification when one cell is giving a signal and the other cell can respond to it. Now, there's always an inducer and a responding tissue. So cell induction requires two types of tissues, one that is secreting the protein and the other that is typically responding. There are situations where it's its own inducer, meaning some cells can release something and that just auto-regulates its own cells. But most of the time, it's one tissue or one group of cells that are secreting a protein that causes another group of proteins to respond. Now, it matters what type of gradient is being sent or what type of morphogen. For example, here we have, these are some samples or examples that you'll see in your book. Here we have just basic ectoderm, or the epidermal tissue that will become the ectoderm or ectoderm components of, in this case, looking probably at a bird. Over here, we have different types of inducing tissues. This tissue comes from the mesoderm where the wing forms. This tissue comes from the mesoderm where the leg forms, and this tissue comes from the mesoderm where the foot forms. So this is the same tissue that is receiving the signal, but each one of these mesoderms are expressing a different morphogen gradient. So what happens is, due to the fact that each gradient is different, is maybe a different protein, the same tissue will respond differently based upon what the inducing tissue is giving. For example, if you put wing mesoderm under this, it will form into wing feathers. If you put thigh mesoderm under here, it'll form into thigh feathers, which are different. And then, of course, if you put foot mesoderm under here, it'll form into the claw in this regard. So even though this tissue, it is fully competent to become any component of the ectoderm of the organism, it matters what the inducing tissue is. Okay? So this is the factors The reason why each one of these is different because they're expressing some different myriad of proteins or combinations of proteins that do that. Now, in some cases, you have what's called reciprocal induction, where one induces one, and then the other one in turn induces the other. So you have this crosstalk between the tissues. It's not always just one's the inducer and one's the responder. Sometimes this tells us what to do, and then in turn, this one comes around and tells the other one, okay, now you do this. Okay? And that kind of falls into line with a concept called sequential inductive events, which just means tissue, as it develops, sometimes goes through this stepwise process that does matter in terms of its overall development. Meaning you can't skip steps. You have to first give it this protein, and it does something. And then you give it another protein, and it does something, and so on and so forth. It can't respond to the second protein until the first one's done its job. So sequential induction events, that's really how most things develop in you and I. It's never just one type of induction. It's this, and then it's this, and then it's this. And that's ultimately what caused these tissues to go down their particular fate, is multiple inductive events that develop the system overall. It's a very complex process. Now, here's an interesting thing. A lot of the morphogen gradients that we're going to talk about today, as well as their subsequent signal transduction pathways, are almost universal in some organisms, meaning we and other vertebrates and even some invertebrates use some of the same proteins to pattern the brain or to pattern the ectoderm or whatnot. The difference is the genetic capacity of those tissues to respond to that. Now, what am I talking about? Well, here's an example where they did a transplantation experiment. They took a newt and a frog, which are similar or closely related to one another in their genetics, and they took the tissue from the frog and put it where the newt's at, or they did the opposite. They took it from the newt and put it in this area where the frog was at. Well, what ended up happening was the frog tissue tells this region it's supposed to become, well, in this case, it's supposed to become the suckers that are on a frog tadpole. Well, in this regard, the newt responds in kind to whatever the frog is inducing. Well, over here, the frog has these newt balancers because that's what the tissue knows to be able to respond to. So even though they both have the same inductive tissue, there's only a limited capacity on what the tissue is supposed to be able to do. For example, here in the newt, normally under this signal, the tissue would respond and become balancers. But it becomes only what it knows what it can become, and in this case, it's the frog suckers from that tissue. So the long and the short of this experiment is showing you that you may have the same inductive tissue, say that this mesoderm tells the ectoderm to become a feather, but if we don't make feathers, we're not going to make feathers, we're going to make skin, you know, under those same conditions. There's a limited capacity to what this. It's not going to say, hey, you're supposed to become feathers, so make feathers. There's no genetic capacity in those cells to do that. But they will respond in whatever capacity they know how to respond. So we can't really generate feathers in our skin because we don't have the genetics to do so. But we would respond to the same signal, and instead of producing a feather, we would produce hair or something of that nature. Okay, that's kind of the idea behind here, is that the inductive signals are the same between species, but the tissue has a limited capacity, depending upon the species, on how it's going to respond. All right. It'll respond as it knows how to in its genetics. Okay. Last types of interactions before we move on. Instructive interaction is ultimately, this is where a lot of the conditional specification comes into play, was that you have to have, the cells are periodically telling these other cells what to do. Okay? They can't respond autonomously. They don't develop autonomously. In order for the genes to be turned on, it has to receive this signal from the inducing tissue. So instructive interaction is one of those things where the morphogen gradient must be present in order for the cells to be able to respond. However, there comes a point where the tissue starts going down this process of specification, and it has everything it needs. It's almost autonomous. It just needs the right environment to finish off. So permissive interaction is one of those things where the tissue has already gone through those stages of induction, and it's almost ready to be finished off. It doesn't need any more instruction from any other tissues. But you still have to keep it in that environment because there are other factors. Maybe there's a extracellular matrix that the cells need to be able to maintain that. Maybe they need to be in contact with other cells. They don't necessarily need an inducer. They just need the appropriate environment, which typically means that they're touching other cells and they've got they have the same conditions that they need in order for that. So it's kind of splitting hairs, but these are kind of different levels of specification. Instructive is the earliest stages. Permissive is the late stages of specification where the cells are almost committed, almost determined to be what they are, and they just need to be maintained in that environment, and then they're ready to go. This is just to give you some basic concepts of the complexity of induction, of conditional specification, where cells, you can see it's not just, hey, throw some morphogen gradients. There's a lot of things. Is you induce me, I induce you, I can respond this way, you go through the sequential. That's really all I'm after here, is that you understand some of the complexity here. So let's go into some of the things regarding cell signal. Let's get into the actual morphogen gradients, the signaling, and what role they play in development. Okay? And you're going to see these signal transduction pathways throughout the entire semester. You're going to see sonic hedgehog. We talked about competence. Okay? Remember, competence is actually the more competent a cell is, typically the more pluripotent it is, meaning it has the ability to bind to and respond to the various tissues. Now, if you look at the human body and you look at all of our tissues, each tissue has a very limited level of competence. What am I talking about? Well, we release hormones all the time, but the hormones will only affect those tissues that are actually expressing the receptor for that hormone. So if we have a growth hormone, only the cell, well, a lot of the cells in our body respond to growth hormone, but only the cells that have that particular receptor will respond in that regard. So you can just, this is one of the things about development, is you can send all these proteins out into the ether, so to speak, or into the area, and only those cells that have the receptor to respond to that will respond. And that's where competence comes into play. And that's built up over time, over the earliest stages of induction that start telling them to express certain receptors. So let me show you how this works. So let's say this is the inducer right here. It throws out this triangle-shaped protein. And notice this cell doesn't have a receptor that will bind that properly. So guess what? It doesn't respond. This is what we would call non-competent. So non-competent cells are cells that cannot respond to a morphogen gradient, to some particular factor. Whereas competent cells are cells that can respond. So competency is relative. If we say a cell is competent, you have to say, what is it competent to? Okay, so competency is relative to the actual inducing signal. This cell right here is the inducer. These are the ones that respond to it, the responder. Okay? So here, what happens, and this is what we're going to go over for the rest of today's class, is when the protein targets a particular receptor on the surface of the membrane, then it starts this cascade of events in the cytoplasm that typically turn transcription on. That's the ultimate goal here, is that it causes new genes to be transcribed and translated, and that in turn causes this cell to start acting differently, creates new receptors that then might respond to a different signal, you know. So that's really what this is all about, is that in the earliest stages, the cells will start turning on various receptors, and certain cells are competent to certain inductive signals. Others are not competent to other inductive signals. Here's an example of how this works. Eye formation, very complex process, but typically there is a tissue that is necessary to cause the lens to start forming, the induction of the lens by the optic vesicle. Now, if you take that tissue and transplant it to, say, this lower region of the embryo where the trunk is at, due to the history of this tissue, it doesn't have the receptors for that morphogen gradient. Over here, if you remove the actual inducer, notice this tissue right here, it won't actually form a lens. There's no signal being produced, and so the responder can't do anything. Again, conditional specification. This tissue must be present for this to become the lens. Otherwise, there's no signal for it to be induced to have that. If you take some other tissue other than the optic vesicle and put it in there, it shows that because it doesn't have the same morphogen gradient, it still won't induce it to become lens. So they did this because they wanted to see, well, what if there's just tissue there? Let's just replace it with a different tissue, and the mere nature of having tissue there doesn't cause that to become the lens. It has to have a very specific protein or proteins that are being secreted by this tissue for that to develop into the lens of the eye. We've talked a lot about transcription factors, and that's what we're going to focus on here, is